Now, after the introduction of the viruses, different diseases which are produced by the viruses, now we will study the structure and formation of the viral particles. As we have already studied that virus is composed of some viral particles which can produce and which are the basic made up of proteins and lipid bilayers. So now we will study in detail that what kind of the particle, just like we see the capsid or the envelope, or what kind of the things which our virus is made up of, and we will study them just one by one, and its structure and function of those particles one by one. First, in the slides, we will study the viral capsids and envelopes. There are two kinds of the virus particles which are very important, which are the most important, which are we will study them. Then one is the viral capsid and other is the envelope. The capsid virus particles contain a viral genome packed in a protein coat called capsid. There is a protein coat which surrounds the genome of the virus. The viral genome, as we have already studied, can be RNA, viral genome can be DNA, viral genome can be RNA interaction. So this genome is surrounded by the capsid, by the protein coat, and this protein coat, which called it capsid, we will study it in detail and we will see the pictures. And second is the envelope surrounding the capsid. Some of the viruses contains that in this envelope, and most of the viruses contain this envelope, which is along with this help they are able to protect themselves, and it can help the capsid and the genetic materials to help them. And similarly, most some of them don't have this envelope. It composed of the lipid bilayer that contains the viral proteins. These viral proteins derived from the host cell membranes. So the host cell membranes provides the viral proteins. These viral proteins made the envelope which surrounds around the capsid. So these two kinds of the viral particles are very important, and we will study them and its functions, what they do in the viral life cycle. The capsid and envelope play very important roles in the viral infection, how the viral infect the host, and how it will interact with the different kinds of the host, how it will use the host machinery for its life cycle, for its life, including the virus attachment to the cells. First of all, the virus, for the infection of the virus, it has to attach with the host cells. Then it will enter into the cells. It will be also the release of the capsid contents into the cells. The capsid contents are the genetic material of the cells, and then packages of the new formed virus particles. The genetic material of the virus will be released into the cells, and the cells will used for its genetically replication. The virus will use the cell's machinery for genetically replication. The capsid and envelope are also responsible for transfer of the viral genetic material from one cell to another cell. And these structures also determine the stability characteristics of virus particles, such as resistance to chemical or physical inactivation. This is most important object of any virus, that whether the virus is resistant to some chemical or physical inactivations or not. Because from human point of view or from the host point of view, we need to control the viruses in different ways. We need to kill the viruses, we need to make them deactivated, just like we are using in the vaccines, or we need to control the viruses, in other words. So in this way, this is very important property of the viruses, that with whether they are inactivated by the physical or chemical way, and these are the make it clear with the help of the capsid and its envelope.